Hello, this is Benjamin Verseman. I will be covering phototransduction in the rod cells for Michael Barton's Biology 201 class that meets Wednesday and Friday at 11. To begin our analysis into the human eye, we will first take a look at the basic construction of it. As we can see in this picture, the eye is a roughly spherical-shaped, fluid-filled chamber where light enters through the pupil, located in the front, then impacts upon the retina, located in the back, where information is then processed by the optic nerves and sent to the brain. The rod cells, which we'll be looking at in this lecture, can be found mostly along the outer edges of the retina and are responsible for peripheral light sensitivity and for low-level vision. A point of interest that must be brought up when analyzing the flow of light and information in the eye is that the eye's focal length causes it to actually flip images upside down when projected on the retina. Take, for instance, in this image we have a small stick figure man standing, and when the light waves travel through the cornea, they actually create an image upside down on the retina. This information is then taken and processed by the brain and image corrected so that the image appears right side up. To begin our in-depth analysis of how light energy is converted into neural impulses, we will be looking at the basic construction of a rod cell. A rod cell is a long cylindrical cell that in the upper regions contains many disc-like structures. These disc-like structures are filled with a chemical compound called rhodopsin, which is very key in the process of converting light to neural energy. Another interesting note is that rod cells are depolarized when there is no light stimulus being placed upon them, and they become hyperpolarized when light impacts the rhodopsin. Rhodopsin is an interesting compound in that it is made of two different proteins, opsin and 11-cis-retinal. When no photons are interacting with the rod cell, the 11-cis-retinal and the opsin are strongly bound to one another to form rhodopsin. To understand the changes that 11-cis-retinal undergoes when a photon strikes it, we will be comparing cis versus trans. In chemistry, a compound is considered cis if two of the dominant groups are on the same side of a double carbon chain, while in a trans, those two groups are on opposite sides of a double carbon chain. This leads to a bent shape versus a straight shape. This relationship of cis versus trans becomes important when we look at how a photon interacts with 11-cis-retinal. When a photon of light strikes 11-cis-retinal, it undergoes an isomeric shift and converts into all-trans-retinal. This change in shape results in all-trans-retinal no longer being able to bind to the opsin complex and ends up decoupling it from the protein. After all-trans-retinal detaches from opsin, opsin becomes capable of activating a G protein called The transducin protein consists of three smaller subunit proteins, known as alpha, beta, and gamma, and a bound GDP, which is guanosine diphosphate. When opsin activates the transducin protein, it causes the alpha subunit of the protein to break off, as well as decoupling the GDP from the alpha subunit and bonding a GTP, which is guanosine triphosphate. This newly liberated alpha subunit of transducin, with its bound GDP, goes out and finds another protein in the eye called phosphodiesterase. Once two alpha subunits of transducin have bound to phosphodiesterase, the blocking units on either side are removed and phosphodiesterase becomes active. The now active phosphodiesterase proceeds through the cell trying to find cyclic GMP, which is cyclic guanosine monophosphate. Once the phosphodiesterase has bound to the cyclic GMP, it converts it into regular GMP. The result of all this phosphodiesterase activity is that cell cyclic GMP levels decrease. As intracellular levels of cyclic GMP begin to decrease, the normally open sodium channels begin to shut, due to the fact that they are held open by cyclic GMP. Although the sodium channels have shut, the voltage-gated potassium and calcium channels are still open. This continuing flow of potassium results in the cell hyperpolarizing and becoming more negative. Once the cell membrane reaches a potential of approximately negative 70 millivolts, the calcium channels shut. This stops the flow of calcium into the cell. The result of this calcium stoppage is that vesicles containing neurotransmitters can no longer bind with the rod cell's cellular membrane and release the neurotransmitters into the gap. Once the rod cell is no longer being exposed to light, a number of cellular proteins begin working. A protein known as guanylyl cyclase is responsible for converting the GTP that is bound to the alpha subunits into cyclic GMP so that the sodium channels can be reopened. Another protein, known as protein kinase G, is triggered by the increasing cyclic GMP levels and phosphorylates many different structures within the eye. Finally, numerous sodium, calcium, and potassium exchange pumps are responsible for re-establishing membrane potentials.